I'm going to show you BioJade. I'm sure many of you have seen this work already. It's actually pretty old stuff. I had to dig it out and reconstitute the parts database. For those of you familiar with BioJade, BioJade actually uses the same parts data model that parts.mit.edu uses. They were created as one, and now parts.mit supports iGEM, and BioJade has sort of been on its own. So let me just restart the program. So when you open BioJade, you have the ability to select the library of parts that you are getting your information from. Right now I just have the old data connection from ROS, which was the original MySQL server that ran the parts database, but now it's locally stored here. So, the resolution is not. Is there a way to change the resolution?
John, I think we connected at like 6.4 earlier.
Get this a little better. That's better. Okay. Now we can actually see everything. So BioJade presents several ways, aspects of looking at your parts. We have a schematic view, which shows the circuit layout of a part or of a design. Functional network, which shows the connectivity of both the DNA and the various proteins that are interacting in the system. A DNA level representation, this one's not filled in, that shows the actual DNA along with the features of your system. The ability to submit your simulations, your systems to various simulators that you register with BioJade, and an icon representation that you can specify. You can click on this and import a picture and specify terminals on your design so that you can specify the input and output to use that part in a larger system. Let me just...
Is that in turn recursive? Can you then pull that into something else?
Yep. So we can design a new part, describe it, and you can specify sort of generic class of parts, and we actually talked about yesterday how you define parts families, and right now there's only a limited set here, but you will have the ability eventually to define more exotic parts. So, kill this other one. So right now you have a blank slate. On the left-hand side, you have the ability to select various types of parts. So we have reporters, inverters, OR gates, NOR gates, sender and receiver cells, or sender and receiver parts, NAND gates, and pop source. So let's build a relatively simple system. Get a reporter here, get an inverter, or two, and pop source. And with the wire tool, you can wire these together. So select the terminals, put them together. You have a design, you can move it around, and if you click on the details here, you can see a transfer curve. This is just a demo, but this is the sort of thing that you would gather from the parts database. This would be data that you've collected. Just more for an inverter, but that was just in the demo. The cool feature of BioJade is that you can push this compile button. Now, there's no information that you put here at all as to how this design is going to be implemented. If you push the compile button, what BioJade does is it goes to the parts database, and for each of these types of part prototypes, it grabs a list of parts that could possibly implement that prototype, and it figures out which ones are compatible based on signal levels, should you have that data, and also based on incompatibilities. So if parts internally contain various proteins, it keeps track of these and which ones are incompatible. And it gives you a list of the parts that you're using. It's up there. And it shows you for each part which one has been allocated here. So this is C0460, which is one of the fluorescent proteins. From that, it generates the functional network view, which basically just shows you how the DNA is laid out and which proteins affect the other ones. So you can see this reporter protein, there really isn't anything affected by it, while the regulatory proteins that are from the inverters are affecting their promoters. And while this information should be pulled from the database, you can set the degradation rate of various species in the reaction network. So you can also change the ribosome binding site and tweak it that way. And when you save it, it's supposed to generate the DNA. There's something not quite right about it. There we go. And this generates a DNA layout, so it shows where the various features that you've designed are on the sequence, and it shows you the entire sequence of the part that you've designed. You can take that design and you can simulate it on various simulators, and I've wrapped a couple of them. Stochasterator, which Drew Endy wrote several years ago when he was a postdoc, and Tabasco that Sri Kasuri and Jason Kelly wrote at MIT. So these simulators are hosted either on a server that you have somewhere, maybe even a supercomputer, or in this case, just local server. And you can execute the simulation, set the simulation-specific parameters. So I haven't run it a few times. And if it was working properly, it would show you that the simulation is currently running. But what it does is it takes those simulation results and forwards them to the parts database. So it's totally asynchronous. And if you have a viewer that can view these simulation results, instead of just viewing it in the BioJade workbench, you can see it in the parts library. So all of these are stored as associated files with the part that is your design. And once you have something you're happy with, you can package it as an icon, mark the terminals, say this is a delay switch with a reporter, and then add it to the parts list. And of course,
like the Tabasco simulator, don't you need a lot of details?
so there are a lot of parameters that it pulls from the parts database, if those were existing. Right. And actually, as you were noting before, the real lack of data is a real problem for actually making any of this meaningful. BioJade was sort of conceived with an eye to the future 6 years ago. We said, oh, yeah, these synthetic biologists, they'll come up with data eventually, and there's actually still very little that we can use to actually make this useful to anyone, which is unfortunate. But I think the ideas are still powerful because it shows you that you can take DNA and put it together from a high-level representation, which is something that biologists really don't like to even think about or consider.
Can you save this as an uncompiled design and then use that in something else and then compile that. So you might have one compilation of this design that uses a certain set of parts, but then in a different context, you
Sure.
already used that part, so it picks a different one for it or something like that. Or
Yeah. And you can actually select the actual implementing part from a list. Like if you want to
Right,
change
you
it,
wanna
if you want
do
something in
by hand.
particular, right?
But so you could save this
Take it.
as all these DVAs, CO 460, but then if you were to try to use it in some other system that also used VBA CO 460, would you be able to change it out? I mean, would it automatically change it out if you hit compile, or would it? Or
So if you specify something, it won't change it out. But if you don't, then it doesn't care. So you can go through. There's a long list of inverters.
So, like your inverter has a transfer function, and that's what you use to model it. Is that how it works?
Yeah.
And
Well, that's for some levels of simulation. You have kinetic parameters that you can use for other ones. So unfortunately, BioJade is less useful without the database, but I think it's still a nifty tool.
Wait. Are you included? Thank you.
Okay.
Never
Wait.
mind. I prepared this question, I just forgot what it was. We'll come back in a second.
Jonathan, can you open the Open Wetware Software Tools page?
Or software tool.
Computational tools.
So I guess we have about 10 minutes, so this is kind of the discussion. What I'm asking Jonathan to do is open the Open Wetware Computational Tools page, and this will show folks kind of what's out there. And the first thing that comes to mind for me, coming new to the community, is there's a lot of people doing a lot of interesting work, and what we should be doing, in my opinion, is finding ways to make the tools work together and work in complementary ways. So I can already see what I presented to me is very in the lab, bench-based, kind of making parts, getting them to work piece. And then we have these simulation tools that maybe once we get parts start to get this characterization sort of developed, maybe then that helps. Clotho could help populate databases. Then the simulation tools could pull from those databases and use to simulate and come up with a flow. So what I'm going to try to do, based on the tools on this Open Wetware page, is to try to get folks that are interested to show me their design flow at a very high level. And maybe come up with a picture for the community of where all the tools are playing and where the connections might make sense. And if they are going to make sense to have connections there, what should the handoff be and what kind of format? So maybe to Tinkercell, maybe what I do is populate a registry and give it an XML file to populate the parts. And then a plugin in Clotho also produces C code for a particular routine. And then it is run. And that C code is parameterized or created in such a way that's specific to the part I just created. And then we can come up with a flow to tie BioJade and all these tools kind of together. I mean, again, so I didn't show in my presentation the actual RTL flow for ASICs. Again, I don't want to do the CAD, the bio thing, but if you look at it, it's very piecemeal. And the reason it is that way is because of how it evolved kind of bottom up, and different companies do different things well. And what we've been successful in is getting tools to talk to each other. There are only a couple of companies that do the whole flow, or a company like a place that has a foundry like Intel will have to control that flow because a lot of that is their value added for their foundry and things like that. But in general, CAD companies need to work together. And hopefully the same thing will kind of work with biology and that.
Is this
It's
the...
always the bottom. Up a little bit. A little more. Very, very, very top.
I think this page, last time I looked at it, had most things represented. I don't think Tinkercell's on there quite yet. But,
new version of Athena.
yeah. So.
You should put this somewhere more prominent than like Ross.
Well, yeah, we just kind of, it took us a couple of minutes to find it, so perhaps it should be moved someplace a little bit more public. And I think the other thing, again, Berkeley bought the whole open source movement. I'm a big proponent of that. And so Clotho is open source, and I believe are your tools as well. And
BioJade
so
is open.
BioJade. So I don't know. I can't speak for the other people, but I know I very much welcome people to want to work on it, and that's why I'm trying to create the plugin environment. It's not my goal to create something and have it over at Berkeley and say this is ours, and the whole point is to get people collaborating. One of the things we talked about briefly at the standards meeting yesterday is that if you're writing code, the most gratifying thing really is when people use it. And so we need to create a user base. And that's also how we find, it's not a very robust software testing methodology, but it's an effective one, that the more users you have, the more bugs you find, the more feedback you get. So really this is only going to work if people start using it, and people are only going to start using it if we respond to what people need and what's out there, and listen to folks and make it open source and give the community a sense of ownership. So
I think you have the added benefit of working with Chris.
yeah.
He's telling you what he actually needs on a day-to-day basis. And when we did BioJade, it was sort of in the high level, this would be really cool if we had this tool, and it wasn't designed with an eye towards usefulness. And it turns out the parts database is much more useful to people than BioJade at this point. Maybe 20 years that'll change.
Okay. So I have a much more specific question, and then maybe we can return to the high-level overview, which I think is very important because it's an efficient way to explain to everyone what's available, and make tools like Jonathan's BioJade less obscure. It shouldn't be sort of forgotten. There are a lot of good ideas in it. But my question, John, it relates to something I think you asked earlier, which is about the crosstalk issue when you're designing a gene circuit with multiple control elements. Everyone always brings up, well, if it's a black box, I'm concerned about using it because it can't really be a black box. If there are incompatible transcription factors or something, or the same one. So, John, it's interesting that BioJade sort of knows that from some sort of interaction table.
Right.
And I'm curious if in SuperPubble or whatever we start to build on top of it, whatever representation standard for talking about BioBricks, can you maybe spend a second and think about and elaborate how we might standardize describing those reactions? I mean, maybe this exists already in a database somewhere, but it would be great. I don't think anyone's doing anything to annotate those interactions consistently, and it's probably very important.
So BioJade uses a parts incompatibility list that maps between different parts. What you'd probably want is a general incompatibility list between protein accession IDs, something like that. But then again, that would have to be at the family level, because if you have one-off versions of those proteins, they'd still interact, right?
Or if or the Athena, what it used to do is, which was like previous version of Tinkercell. It's kind of odd. It didn't really use anyway. What it did is it got all the transcription regulations from a database, I think one of the largest transcription database it's from EcoCyte. It's called RegulonDB.
Okay.
So that has data on what protein binds what transcription, what binding site or what promoter. And so if you have all that list, and then you you use a new protein that, and if you could debate. So if you have that information, and then suppose you use a new protein that binds to something that exists in your circuit, then the software can say, warning, this binds to a site that's already in your circuit. Is this intentional? Something like that. So if you have a database of all interactions, then I think that can.
Is that sufficient, or is there something else we should do to encourage people to use it in a consistent manner? I mean, is it enough to just say, well, go use RegulonDB, they've got an API or whatever, or a common way of representing this stuff, just go talk to them if you're building a tool? Or should we be more intentional about it? Because I feel like this question comes up a lot, and no one ever says, oh, I just checked in RegulonDB.
Well, I think we want to capture that data, but people are always finding out new incompatibilities. There are new systems being created all the time. So if you have that as a snapshot, it may be useful temporarily, but you'd probably want to go back and validate it with the RegulonDB or whatever is keeping track of these interactions.
And there's several. This is one level of incompatibility, right, where you're using proteins that bind to multiple places, and that's not desired.
Right.
Suppose you have other types of crosstalk, such as DNA. You have like homologous DNA next to each other, and they form some secondary structure. Then Regulon DB would not tell you things like that.
Do you think it would be possible to build another computational tool that essentially took two part
Existing database
or two Pubble entries or something, and then magically figured out if they would interact?
Just based on like common proteins and binding sites. That, yeah, that can be done.
The idea would be to separate out that function so anyone could use it. I don't know if that's necessary, but
Actually, that's a good idea.
Yeah, you'd be able to do that. We're also
to centralize it, and also to maybe keep special lists that are BioBrick only or something, or very temporary, that don't deserve to be in Regulon DB but are still sort of provisional crosstalk observations. I don't know.
So we're also working on a tool that may or may not become part of BioJade, but at least separately available to manage secondary structure when dealing with RNA elements in your transcript. So we're building RNA sensing devices, and you want to sort of maintain the structure of those domains, as well as the ribosome binding site just interacting with coding sequences. And that's a general problem that we'd like to manage computationally.
Okay, well, Doug's looking at the clock, but I have one more question, which is maybe everyone that could explain explicitly what the development schedule is like for their program or their tool. I mean, are people directly working on BioJade right now, and if they are, where do other people go?
No one's been directly working on BioJade in 5 years.
But you were just saying that maybe something new for BioJade would be blank.
Maybe.
That's actually a good question. So another thing
I need to graduate.
So I'm not burdened with having to graduate, but I think that's an excellent idea. So I can speak to Clotho. Clotho right now, Alpha is at the website. It doesn't have all the COBOL stuff, so it's somewhat sort of hamstrung in that way, but it's still there to play with. iGEM, there'll be Alpha 2. I'm not calling it Beta because I don't want to call it Beta because it's not that robust yet. But then we'd like to have a real Beta, and I'm reason to call it Beta now. Version 1 because there'll still be some bugs in the spring. And then I'll definitely like to have, by next summer, there'll be the official, hopefully bug-free tested, working in Anderson's lab version of Clotho. And then my trajectory is to go into academics, so I imagine at some point that Clotho will be alive and well, working in some form with other students behind it.
Cool.
So I'd like to. What about your
yeah, antimony is. We hope to release version 1.0 basically in January or February, I think. So, in time for the latest NIH update on our grant.
Tinkercell, hopefully there'll be a alpha sometime this month, and which is basically what you saw. It just needs a few things, and I'll be working on it on and off, adding more functionality. But yeah, I need to graduate also, and that's not my main project.
Great. So let me just end by saying that, as I said, when I go to conferences, everyone is talking about synthetic biology in our community, so we're coming, whether you like it or not. So I think the best thing to do is to take advantage of this opportunity and educate us. So if you see a computer scientist and they say they're thinking about getting into synthetic biology, and we have the questions, about, we say we just want to put logic gates down, push back on them and say, how are we going to deal with these issues? These are the things. Educate us about your community, and I think you'll get better results than trying to, find an aggressive stance. And so far, this community actually has been very welcoming. So continue that, you guys. It's exciting, and with that, get involved. So.
Great.